Now that we know all there is to know about a primary antibody, let's talk a little bit about what are our options when it comes to secondary antibodies. Secondary antibodies are antibodies generated against an immunoglobulin. That means they'll be able to bind to your primary antibody. Matter of fact, they are produced exactly as we described for your polyclonal primary antibodies, except that the animals are immunized with an immunoglobulin. It could be a whole immunoglobulin or a fragment of an immunoglobulin. Because of its polyclonal capacity, the secondary antibodies can bind to several epitopes of the primary antibody, which ensures that we're going to have signal amplification when we use a secondary antibody. On top of that, your secondary antibodies are generated in a specific way so that they will recognize only the Fc region of your primary antibody, which ensures it will not affect the binding of the primary antibody with your antigen of interest. Secondary antibodies can be labeled with different molecules. For example, they can be labeled with an enzyme such as Horseradish Peroxidase, which is also known as HRP. In that case, when it's time to image your Western blot membrane, you're going to treat that membrane with a substrate. If you use a colorimetric substrate, you're going to have a colorimetric Western blot. You could also use ECL, which will lead you to have a chemiluminescent Western blot. You also have the alternative of using a chemifluorescent substrate for a chemifluorescent Western blot. Whenever you're using HRP, you're going to be depending on an enzymatic reaction. That leads to a few challenges when it comes to data reproducibility. If your secondary antibody is labeled with a dye, you can bypass the need of using a substrate before you image the membrane. In that situation, the dye will be excited by a particular wavelength and a detector will capture the light that is emitted by the dye. Skipping the use of a substrate will bypass the enzymatic reaction between HRP and its substrate, which leads to a more reproducible Western blot. In addition to that, if you choose to use a near-infrared fluorescent dye, you're also going to increase the sensitivity of your assay due to the lower autofluorescence that comes from both PVDF and nitrocellulose membranes on those wavelengths. To learn more about the different chemistries that you can choose to use on your Western blot protocol, please refer to our previous Lambda U courses.